Hi, we hope you are all enjoying the summer and ready for vacation. My name is Julia.
And my name is Garima.
Have you ever wondered how animals live in their environment, where they come from, and how humans play into the picture? Ecologists do this for a living.
Today, we'll be looking at the intersection of ecology and math in the real world.
Before we get into it, understand exactly what ecology is. Formally, ecology is a branch of biology that relates organisms to one another and their physical surroundings. In other words, ecology looks at how, where, and why animals and plants exist and interact with each other.
Next big question, what do ecologists do? Well, ecologists are a combination of scientists, math modelers, and explorers. They take detailed measurements about the environment in order to better understand how it works and make sure that we preserve it for future generations. Ecologists explore different environments all across the world and find answers to questions that help them understand the world better.
What sort of environment do animals live in? What makes that environment special or different? What factors contribute to animals' ability to live in that environment? Ecology is dirty, hands-on, and data-driven. A lot of the time, ecologists are out in the field finding answers with a close team of highly specialized individuals. Ecologists solve real-world problems.
Ecologists are responsible for some of the biggest breakthroughs in science and life as we know it. For example, have you heard of Charles Darwin? He's the guy who discovered that all living things change through evolution.
Or you might know marine biologist Rachel Carson, who helped shape environmental activism as we know it with her groundbreaking book, Silent Spring, in which she chronicled how harmful the pesticide DDT was to the environment. In her book, Rachel Carson detailed how DDT trickled up the food chain, leading to the death of birds and other animals.
You might know of Jane Goodall, an important ecologist and researcher who lived with and helped us understand our relationship to chimpanzees and gorillas. She did some world-changing fieldwork and helped us understand how primates can act, how humans would traditionally behave.
And of course, you should know Tyrone Hayes. He's a UC Berkeley professor who researched the effects of atrazine on animals and found that it's toxic for the environment. He is also a well-known advocate against chemical pollution and environmental racism.
Frankly, ecology has given us a lot of things to be thankful for. Studying animals and their ancestors brought us the theory of evolution. Looking at the effects of excess greenhouse gases on ecosystems allowed us to discover climate change. And understanding how food chains work have helped us discover how harmful pesticides such as DDT can be.
One of the first things an amateur ecologist learns about is ecosystems and food chains. Ecosystems consist of a community of interacting organisms and their physical environment. This includes each organism, its species, its habitat and diet, otherwise known as niche, physical landscape, and food chains. Food chains show the predator-prey relationship between different species in the ecosystem. Each step on the food chain is called a trophic level. In order to protect our environment, it is important to know how food chains work and how the organisms that make up food chains are interconnected.
For example, consider the simplified food chain. You can clearly tell the grass provides food for the deer, and the deer provides food for the wolf. All three, the grass, the deer, and the wolf, provide integral roles to the ecosystem. Ecologists call each of these roles different trophic levels. For example, the grass is called the primary producer. Why? Because it provides food for the rest of the food chain. It's the base. The deer would be an example of a primary consumer because it gets food directly from the primary producer. Lastly, the wolf would be a secondary consumer because it gets food directly from the primary consumer, which is the deer. These trophic levels go on and on. You can have tertiary consumers and quaternary consumers, and so on. Of course, food chains can be as complicated as you make them. Food chains become pretty complicated in the real world. A lot of ecologists call such complicated food chains food webs.
However, food chains are, more often than not, delicately balanced, and though they can recover relatively easily from a minor change, like a summer with slightly less rain or a winter that is a little too cold, if one link in a food chain is taken away, it can cause the whole food chain and ecosystem to completely collapse. Ecologists call this a trophic cascade. For example, say the secondary consumer in the food chain, in this case the wolf, is removed. Without the wolves to keep the deer population in check, the deer population increases exponentially. With so many deer, the food that the deer eats, grasses, undergrowth, fungi, bark, become scarce, and other organisms that rely on similar food, such as rabbits, die out. This is an example of a trophic cascade, and it shows how delicately ecosystems are balanced.
Another thing to keep in mind is biomass. Biomass is the mass of the population as a whole. Only 10% of food from the lower trophic level is converted into biomass for the next trophic level. That basically means that only about 10% of the grass each deer eats becomes a part of the deer. The rest is flushed out.
But it's a different story with toxic chemicals like mercury or DDT. These chemicals don't dissolve in water, so any of these chemicals that a primary consumer, like the deer, eats stays inside the deer's body for a long time. This means that 100% of all toxic chemicals convert to biomass as they go up trophic levels.
Think about why this is bad news. For example, consider that each kilogram of grass has 3 micrograms of mercury in it. Each deer will eat around 12 kilograms of grass a week. Simplified, this means by the end of 1 week, the deer will have upwards of 35 micrograms of mercury in its body. Anything above 30 micrograms is lethal for the deer. This gets worse higher up the food chain.
This accumulation of toxins going up the food chain is called biomagnification. This is why pesticides such as DDT can still be so deadly to birds, and how when enough mercury accumulates in fish, such as tuna, it becomes poisonous to us humans.
One case of this is how toxic waste products from factories such as polychlorinated biphenyls can accumulate up the oceanic food chain. When PCBs are pumped into the ocean by factories, they're absorbed by phytoplankton, which are then eaten by zooplankton, which are then eaten by herring, which are then eaten by salmon, which are eaten by large mammals such as orca. As you go up the food chain, the concentration of PCBs in each organism grows exponentially. The orca end up with having a lot of PCBs in their bodies due to biomagnification, which is really dangerous for their health.
Here is another example of how biomagnification can lead to high toxin concentrations in the marine food chain. In this case, though, we're talking about humans at the top, so the toxins that are put into the food chain directly affect us as the species on the top of the food chain eating fish.
How do we quantify this phenomenon? One way is through introducing the biomagnification factor to see how much a chemical accumulates across trophic levels. The biomagnification factor is defined by dividing the concentration of toxin in the predator by the concentration of toxin in the trophic level below it. The concentration of toxin for any species can be found by multiplying the total number of prey eaten by the concentration of toxin in each prey.
Let's look at how this works in a simplified case. Say we have a basic food chain with 3 trophic levels made up of 3 organisms: grass, deer, and wolves. There's mercury present in the soil, which is contaminating the grass. Let's assume that each kilogram of grass has zero point 0.1 micrograms of mercury, and each deer was found to have 5 micrograms of mercury, and we're trying to find out the concentration of mercury in a wolf.
In order to solve this problem, let's first define what we know and our own assumptions. We already know the concentration of mercury in both the grass and the deer. We just need to figure out how they relate to each other and how they relate to the concentration of mercury in the wolves. Let's assume that in the food chain, each deer eats 50 kilograms of grass a month, and each wolf eats 2 deer per month. Of course, this will not be true all the time, but if we generalize our model, we can work from there.
Our next step is to define our models. In order to answer this question, let's use the equation for the biomagnification factor along with the equation for the concentration of chemicals in an organism, both of which we learned earlier.
How can we use these assumptions to find an answer? Based on our previous knowledge, we know that each deer has 5 micrograms of mercury, and the grass has 0.1 micrograms of mercury per kilogram. We can find the biomagnification factor for these two trophic levels to be 5 over 0.1, or 50. Now, the lethal amount of mercury for a deer can be estimated to be around 35 micrograms, and we know that each deer has 5 micrograms of mercury, so we're not in the hot water yet. But let's consider the wolf. We can find the concentration of mercury in the wolf by multiplying the number of deer eaten per month, which is 2, by the concentration of mercury in the deer, which is 5 micrograms of mercury, in order to get a total concentration of 10 micrograms of mercury per month. We can go even further in this model and use the average weight of the wolf as 40 kilograms to get a total lethal concentration of 4 micrograms of mercury in an average wolf. So, a concentration of 10 micrograms of mercury is pretty dangerous.
What we just did in our example and all of our case studies is math modeling. Math modeling is the use of math and research to make predictions about the real world. For example, calculating biomagnification, changes in population, the spread of diseases in an ecosystem, are all math models that ecologists use in their work.
The first part of math modeling is identifying the problem you're trying to solve. What's the exact question, and how would you define a successful answer?
The second thing you need to do is make assumptions and define essential factors. What do you need to know to find your answer? Draw and research food chains, food webs, diets, habitats, and anything else you need. Can you assume some things about your factors? For example, in our problem, our factors were the food chain, the diet of the deer, the diet of the wolf, the concentration of mercury in the deer, and the concentration of mercury in the grass. The problem gave us these values. We could have researched the diet for a typical deer and wolf, but chose to assume that each deer ate 50 kilograms of grass per month and each wolf ate 2 deer per month instead. This was reflected in our food chain.
Third, write down all of the equations you would need to solve the problem. We use the BMF and concentration equation, but you might use equations you come up with on your own or something you found online. Then, do the math and get a solution.
Then, try out your model on some new data. Does it still check out? Change it as you need in order to get a good model.
Lastly, analyze your final model. What went well? What went wrong? How can you improve it? Can you use this model for other scenarios? What does your model or solution say about the state of the ecosystem? Remember, there is no right answer. We're looking for creative, well-explained, and applicable solutions.
Today, you'll try some math modeling on your own. Pretend you're an ecologist investigating a recent chemical spill at Lake Quinsigamond, Worcester, Massachusetts. Your team found that because of the spill, there was an increase in mercury concentration at 4 micrograms per kilogram of phytoplankton in the lake. Is it safe to still fish there? Is this dangerous to the environment? If you were to fish and eat a 1 kilogram carp from the lake, the mercury levels in the carp be high enough to be dangerous for you?
You can consider all sorts of parameters such as food chains and food webs, diets and habitats of organisms, what you consider a dangerous concentration of mercury to be, your own math equations, and even cool simulations. If you live nearby, take a look at Lake Quinsigamond and use your observations or previous experiences. Feel free to take this as far as you like. Submissions are due tomorrow before 10 a.m., and you can find the form to submit each problem on the homepage of our website, which is mmjam.github.io. You can find some helpful resources and the problem written up on the ecology page of our website. Feel free to email us at mjam.team@gmail.com. Thank you so much for tuning in, and we hope you learned something. See you tomorrow.
The people you saw in this video are current juniors and seniors at the Massachusetts Academy of Math and Science, a STEM immersive high school located in Worcester, Massachusetts. In collaboration with WPI's Touch Tomorrow, our student-led team was able to organize the entirety of the Massachusetts Math Modeling Jam and bring it to you this year. Inspired by a math competition we all competed in junior year, the MMMJam is just a small taste of the hands-on learning and incredible opportunities that this city has to offer. In fact, it's these kind of events that have inspired all of us to continue learning about science, technology, engineering, and mathematics in college and beyond. If you live in Massachusetts and want to check out more events like this one, be sure to look at WPI, Touch Tomorrow, and Mass Academy at the websites here. Thank you for watching, keep exploring, and happy modeling, everyone!